Good morning, everybody. For those of you who just joined, welcome to the morning session of the third day of the NASA Gregory G. Leptoukh second online Giovanni workshop. This is your host, Jennifer Brennan. So what we are going to do this morning, we are now getting ready to have our second speaker for this morning's session speak. I would like to take a moment here to introduce him. Our second speaker this morning is Dr. Shovonlal Roy, who is a lecturer in the Department of Geography and Environmental Science at the University of Reading in the United Kingdom. Dr. Roy?
Thank you very much, Jennifer, for the introduction. I would also like to thank Dr. James Acker for inviting me for this workshop. It's a great pleasure to be here. So the topic of my presentation is phytoplankton dynamics and time-varying ecosystem indicators using NASA Giovanni data. This research I have been doing over the last few years, and is mainly funded by the Royal Society of London and also by the NSERC of Canada. So as we know that ocean color time series provided by the NASA Giovanni are proving to be very useful for qualitative and quantitative research on oceanic ecosystems. So in this presentation, I will talk about the methodologies and approaches that we have developed recently to understand the dynamics of marine phytoplankton and their community structure using Giovanni time series. So in particular, I will talk about ecological models, bio-optical models, and data assimilation, and will show how these can be used to understand important aspects on plankton dynamics, namely how to understand the phenology of phytoplankton bloom, and secondly, to estimate important biological parameters and how they vary with time related to plankton dynamics. Thirdly, to estimate the time-varying phytoplankton size spectra, which is related to their community structure. And finally, how to compute phytoplankton functional types and size classes from ocean color remote sensing data. So let's start with my research goal. So the two major research goals on using ocean color data for me was to understand the qualitative dynamics of oceanic ecosystem, and secondly, to estimate important ecosystem indicators. And I've used data from Giovanni, and here are some list of the data that I have used. I have used the chlorophyll and PAR, photosynthetically active radiation, from sea whips from 1997 to 2007. I've also used chlorophyll and sea surface temperature and level 3 remote sensing reflectance data from Moody's Aqua for the period 2002 and 2011. I have also used the output from NASA's Ocean Biogeochemical Model, NOBUM, and in particular I used the output on nitrate concentration, mixed layer depths, and biomass of phytoplankton functional types for the period of 1998 to 2007. So let's start with the first topic, which is the qualitative dynamics of phytoplankton. So we know that the seasonality of algae and the herbivore production depends on latitude. And we know from early works of Cushing in 1959, the match-mismatch hypothesis that says that the survival of the fish larvae depend on the availability of food, in this case the algae, and in particular if the timing of larval recruitment matches with that of phytoplankton bloom, then that has an impact on their survival. Now if we look at the phytoplankton bloom dynamics in the global ocean, we see that in Arctic Ocean, phytoplankton blooms once a year, whereas in temperate and tropical region, we see phytoplankton blooms twice a year. This is an observation made long before. But then if we look at the Giovanni time series, so this is from seaweed data 97 to 2007, and what we did is we take a 1 degree cross 1 degree box in the North Atlantic. So these are 10 boxes. The box in the north is around 61.5 degree, and the south is around 43 degree. So you see that in box 109876, you can see phytoplankton blooms twice a year, whereas in the box 61.5, it blooms once a year. So this is very similar to what has been observed long before. Now if we look closely to these two boxes, the box at 61.5 degree north and 43 degree north, we see that the PAR, photosynthetically active radiation, it has a very similar shape in these boxes, whereas the phytoplankton dynamics differ very differently. So the question is, what drives this dynamics of phytoplankton? And to understand that, we developed a dynamical model which we call the energy phytoplankton-substrate model. So this is a model where phytoplankton and substrate interact, and PAR from satellite is an external periodic input. So this dynamical model, when we simulate, it shows the dynamics very similar to what we see in the ocean. For example, at 45 degree north, when you simulate this model, we see that phytoplankton peaks twice a year, and whereas at 65 degree, it peaks only once. So in other words, this dynamical model, when forced by PAR, can explain the two peak and one peak phenomena. However, this model can further be used to compute the timing of spring bloom. So we can see here the timing of spring bloom from seaweed data we calculated for all the 10 boxes. And the right-hand side, these are the timing of spring bloom from the model using different initial conditions. And if we look at carefully, we'll see that the timing of the bloom presented by the model is very similar to what we see from the satellite data. For example, at 45 degree, we see the timing of bloom is around 80 days, and this is very similar to what we see here, and same for 65 degree. So this model can also be used to predict the timing of spring bloom. However, there is something more that the model says, and that is related to the periodicity of phytoplankton biomass. So when we simulated the model, we see that there is a latitudinal band where the biomass of phytoplankton has a periodicity more than 1. And this was quite interesting observation from the model. And then when we looked back to the seaweeds data, we actually found the evidence for that. And this is the evidence. These are two different weeks of a year. And what we plotted here are the histograms of the chlorophyll concentration from one of the boxes. And the blue are for the odd years, and red are for the even years. And clearly the odd years and even years distribution are very different, showing the periodicity is more than 1. So this dynamical model can be then used to understand the phenology of phytoplankton bloom, to calculate the timing of the bloom, and also to understand the periodicity of phytoplankton biomass. Next we move to the quantitative estimation. So in this case, I will give you two examples. One is state parameter estimation, where I'll show you how we can use Giovanni time series along with model to understand the temporal variations of important biological parameters, in this case phytoplankton growth rate and mortality. And the second example, I'll talk about how we can obtain the estimates of chlorophyll series at individual size level. And this will contribute to understanding the phytoplankton functional types from space. The first example is the state parameter estimation. As we know, phytoplankton dynamics are often understood through biogeochemical models. And these coupled biogeochemical models are often, so these biogeochemical models contain constant parameters, and these constant parameters are generally used in most of the cases. However, we know that the dynamics of phytoplankton change in a short time period, and so are some of the important biological parameters. But these biological parameters, for example, the growth rate and mortality, are very difficult to estimate over time and are often not possible due to lack of in situ measurements. So we developed a methodology to estimate the time variation of these parameters. And in this case, we consider a specific area, which is the Canary Islands area. And we consider a simple biogeochemical model, which is a nutrient phytoplankton zooplankton model. This model was parameterized in this area by the authors. However, the dynamics of phytoplankton in this area are influenced by several physical forcing and mesoscale dynamics. So there's a high chance that the growth rate and mortality of phytoplankton change over time, which we don't know. And we also retrieved the chlorophyll time series from Modi's Aqua using Giovanni from 2002 to 2010. And we implemented a data assimilation technique. In this case, we call this a dual state parameter estimation using Ensemble-Kalmer filter. So the data assimilation experiments shows the results as follows. So these are the blue dots are the chlorophyll concentration from Modi's satellite. And we did several experiments with this model. And the easiest experiment was the experiment C, which is no estimation of the parameters, just use the existing one. D is the experiment where we estimated the parameters but didn't allow them to vary over time. And that's these. And A and B are the experiments where we allowed the parameters to vary over time. And A in particular is, along with that variation, we used the state correction of assimilation. And clearly, the black curve and red curve are much closer to the blue dots. And if we look at the errors, we can clearly see the experiment B, for example, is the error, total error is much less than experiment C and D. So in other words, if we can estimate the time variation of these key parameters, then our biogeochemical models are much closer to the observation. Now, because this was a state parameter estimation, we obtained the time series of growth rate and mortality. And these are the time series of phytoplankton growth rate over time. The red curve are the actual estimated values compared with what you see in the dotted line, which is the fixed value. And the gray area shows the range of estimation. And clearly we see the estimated series is very different from the constant series and very different for a periodic input obtained from equations based on sea surface temperature. And same thing for mortality. So mortality rate, again, the red curve is very different from any fixed value or any predefined values. Now, if we look at the time series estimated much more carefully, we see something very interesting. So this is the growth rate of phytoplankton against sea surface temperature. And these are the curves which are known as the function defining how phytoplankton growth change with temperature. These are the Apley curve, and this was later on modified by Bissinger et al. 2008. And the green one is generally used for several biogeochemical models. And we see that the estimated phytoplankton maximum growth rate from our model varies from the others. And in particular, the 95th quantile, which indicates the maximum possible phytoplankton growth rate, it has a very different shape. And actually the slope of this curve is very different from the others. The slope is higher than the others. And if we look at the slope, we see that at 95th quantile, the phytoplankton growth rate has a higher slope. So in other words, this means that the sensitivity of the maximum growth rate to sea surface temperature is higher than what has been predicted by the classical works. So in other words, at a regional scale, phytoplankton growth rate may be more sensitive than we actually thought to be. Next, we move to phytoplankton community structure. So if we look at the phytoplankton community, this is a very diverse community, including many, many species. And these can be distinguished based on various criteria. So generally, we define them based on their biogeochemical functions, which are called functional types. This can be distinguished based on their biogeochemical function, also based on their taxonomic groups. For example, diatoms are very different from cyanobacteria, but also they can be distinguished based on their physiology or cell size. And in this case, often they are classified in three major size classes, which are picoplankton, nanoplankton, and microplankton, with their usual definition of less than 2 micrometer, 2 to 20, and more than 20 micrometer. However, phytoplankton cells within each size class also vary, and it is very important to know the contribution of phytoplankton biomass at individual size level. The reason why individual size is very important are many. So individual size determines phytoplankton primary production rate. This is related to nutrient availability. It's related to light absorption properties, bio-optical property of the water column, sinking loss. Also, it modulates the response to grazing, and definitely is related to the energy flux and ecosystem stability. So it's very important to know the contribution of phytoplankton biomass at individual size level. However, it's not easy to get the individual size from phytoplankton chlorophyll. So from satellite, we get total chlorophyll, and it's a difficult job to split into the biomass of individual cells. So we developed a method of data assimilation where we retrieved the time series of total chlorophyll from CWIPs. This is from 98 to 2006. And we also retrieved other variables, for example, the time series of nutrient concentration, mixed layer depth, local known irradiance, local day length, all this information. And then we developed a size structure dynamical model. And this model is different and new because the parameters of this model are explicitly determined by the cell size of phytoplankton. And then we implemented, again, a data assimilation algorithm. In this case, we used a sequential data assimilation to assimilate all these variables in the model. And so we split the total chlorophyll into the chlorophyll at individual size. I showed here 10 size. One is starting from 0.45 micrometer, and it goes up to 40 micrometers. And then we can obviously pull together to get the large cells, medium cells, and small cells. And clearly we see that the time evolution of chlorophyll concentration at each size level is very different from the others. Now, once we split the chlorophyll into the chlorophyll at individual size, we can estimate a very important property which is called the size spectrum of phytoplankton. And the slope of the phytoplankton size spectrum is a very important ecosystem indicator because this is related to the energy flux and dynamic stability of ecosystems. And once we calculate the size, chlorophyll concentration at each size level, we can then calculate the slope of the size spectrum, which has been plotted here for this location. And we can see that the slope of the size spectrum varies between the years, and also within a given year. And the total variation of the slope from their mean value is within plus minus 15%. And we can compute these from any location. And so what we did was we retrieved the data from AMT crews where these values were measured in 3 AMT cruises. It's not easy to get the phytoplankton size spectrum data. However, in these AMT cruises, the data were available, and we saw that the estimates from our computation is very similar to what has been measured in the AMT. So basically, data assimilation with this dynamical model can be used to estimate this important ecosystem indicator. We then compared our estimates at different size level to the existing phytoplankton functional type models. In particular, here we compared our results with the NASA ocean biogeochemical model. And what we did is computed the total chlorophyll for the large cells and compared it with the concentration of diatom from novum. Now we see that in many places the diatom peaks are matching with those for the large cells, but there are other times when they are not. Similarly, for the small cells versus the cyanobacteria concentration from novum also matches in many places, but not in others. So this result suggests that when we estimate the chlorophyll concentration strictly based on size, they may or may not match to those with a given taxa. And this result can be understood from the fact that sometimes these taxa may appear as large or small cells. Diatoms are known to appear in varying cell size. And in the final part, I'll talk about bio-optical algorithms and how we can use to understand phytoplankton community structure. So as we know, the specific absorption spectra of phytoplankton are in the visible range, has a very specific shape here, so 2 peaks. And these spectra vary for different taxonomic groups. So the spectra have implicit information about the taxa. Moreover, these spectra also have important information about the cell size. And in particular, the peak at the red band here is very important because this peak has very explicit information about the size of phytoplankton cell. And we used this peak, the red band, and developed a bio-optical algorithm to compute the average size, cell size in terms of diameter of a phytoplankton population which we consider to be homogeneous. So in this case, the red curve here shows the bio-optical algorithm function that connects the in vivo specific absorption coefficient of chlorophyll A at the red peak against the diameter, average diameter of the cells. And we see that this decreases with cell size according to this curve. But now, if we use this model to compute the cell size for different taxonomic groups, we see that there's a large variation, for example, for diatoms. Diatom cell size vary a lot, and also other groups also vary. Now, this model then can be extended to the case when we consider phytoplankton as a mixed population, and assuming that they follow a specific size distribution, in this case a Jung type distribution, that homogeneous model can be extended to compute the exponent of phytoplankton size spectrum, which is xi. And this curve shows how the exponent of size spectrum vary as the specific absorption of phytoplankton at the red band change. And the gray area here shows the range of prediction from this model. Once we calculate the specific absorption spectra, and then once we calculate the phytoplankton size spectrum, this can then be related to compute the concentration of chlorophyll at any given size class. And in this case, I show here for 3 specific size class, pico, nano, and micro. So these 3 curves shows how we can connect the size spectra exponent with the biomass at 3 size classes, and in other words, how we can connect the specific absorption spectra with the chlorophyll fractions at 3 size classes. And obviously, the blue curve is the pico fraction, which increases as the specific absorption spectra increase. The micro fraction decrease, and this is the nano fraction. This model then can be used to compute the size classes from satellite. And here we used again the Giovanni Modi's Aqua data. This is a particular image of winter 2011. This is the chlorophyll concentration, and we computed the specific absorption coefficient of phytoplankton at the red band using an IOP, inherent optical property algorithm. And then we applied our model to compute the exponent of phytoplankton size spectra. This is that, and the average diameter of the cells present. And you can see that the exponent of size spectra excuse me, is greater than 3.75 in most of the oligotrophic gyres, and is less than 2.5 in most of the coastal waters. And same thing for the average cell size is less than 0.3 micron in the oligotrophic gyres, whereas in the coastal water is more than 1.25 micron. We can further compute the percentage of picoplankton, nanoplankton, and microplankton, and we can see that as we understand from the spatial distribution of phytoplankton, the picoplanktons dominate the oligotrophic waters, and we can see it can go 70 or 80%, whereas less than 20% are present in the coastal waters, and the opposite is for microplankton. It's a large fraction in the coastal water and very less in the gyres, and nanoplankton has a distribution like that. Now, because this is a bio-optical algorithm, which is a deterministic model, this can be used to also compute the uncertainty in our estimation. And this we calculated the uncertainty in estimation of all those quantities, and we can see that the percentage of uncertainties vary spatially. However, there's one important feature of that, and that is the percentage relative uncertainty in estimation of any of those quantities are always less than the percentage error in the input data. So in other words, if the data error is less, we'll have much finer estimation. And that brings us to the conclusion. We saw that the seasonality of phytoplankton shows complex dependence on latitude, and we could develop a dynamical model, simple dynamical model, that includes annual forcing based on remote sensing, which can be used to capture the bloom phenology and also can predict the timing of the bloom. And we saw that state and biological parameters of phytoplankton dynamics can be estimated from ocean color using data assimilation, and also the size resolved biomass fractions can be estimated from ocean color. And we have seen that if we estimate the biological parameters as a function of time, then the time evolution of this growth and mortality rate may be very different from the predefined values, at least in a regional scale. And we have seen that bio-optical models can be used easily to implement for computing cell size, size spectrum, and size classes of phytoplankton, of course using specific absorption spectra which can be derived from the ocean color, and the uncertainties in functional type estimation or any of those quantities would depend mainly on the input satellite ocean color. In other words, if we have a better quality satellite data, that would produce finer results without retuning any of these parameters of the model. I would like to thank NASA Giovanni for making this data available for this research, and also to the principal investigators who have provided the data. And obviously the reference for this is this I have used. And here are the papers that I was mentioning during the talk. And I would also like to thank the co-authors of this paper. And here's my contact information. I can be contacted using any of those methods. And I'll be very happy to hear from anybody who would like to use any of the methodologies or algorithms I've talked today. And thank you very much for listening. Thanks.
Okay, thank you, Dr. Roy. So what we will do next is we will move to the question and answer period for our speaker, Dr. Roy. If any of you have any questions for him, please do type those questions into the Q&A pod located on the right-hand side of your screen. Also of note, we do have his presentation uploaded below in the file sharing pod, if you can see here. In order to download that file, all you would do is use your cursor to highlight the file, at which point you will have an opportunity to download. So I do see we have some questions, so why don't we go ahead and get started here. Pardon me. Okay. It's quite long. Okay. So I'm going to go ahead, Dr. Roy, and read the first. It looks like there is a combination of questions and comments here from Adonis. Okay, as we know, phytoplankton population are essential, as with plant and sea life it supports. In the case of existence of biotoxins associated with the presence of nitrates and other pollutants in the water, how do those contaminated marine life do away with such a threat? Do those contaminants go away naturally in a few days? What are some human interventions required to ease its impacts on the food supply? So it sounds like there are a few questions, and that in the case where biotoxins exist, associated with the presence of nitrates and other pollutants in the water, first part of the question is, how do the contaminated marine life do away with such a threat? One. Part two, do these contaminants go away naturally in a few days? And part three, what are some human interventions required to ease the impacts on the food supply?
Well, I think this is quite a bit of general topic and probably a little bit away from what I have talked today, but still let me make a few comments on that. I think the biotoxins has a natural degradation property, so they would go away for sure. But there are complex interactions within that. So for example, during toxin blooms, we see that some of the toxic species grow and there's a lot of biotoxin in the water. However, once the bloom degrades, then gradually the toxin level will also degrade over a few days. So in other words, it's a kind of regulation on themselves. However, that biotoxin has severe impacts on the ecosystem, and that impact would be maximum at the time when the toxin is present. However, the human intervention, well, it depends on how we intervene these things. Sometimes it's not easy, and these in coastal oceans, often the toxin-producing phytoplankton create like the harmful toxins that have impact on human health. So, I mean, it's basically a kind of open question of how we can intervene those Obviously, biologically, one may think that if the toxin-producing species do not bloom, that's the best idea. But that's not always possible because of the complex interaction within phytoplankton community. If there are many species and the competitive exclusion principle suggests that one of the species will grow suddenly and different nutrient and other conditions allow some of the toxic species to grow periodically or sometimes even randomly. So it's kind of an open question of how we will be able to control the bloom or how we can intervene to control the toxic blooms.
Okay, thank you for your response, Dr. Roy. Our next question is from Cecile Rousseau. She said on slide 15, would you like me to pull your slides back up to address this question?
Yes, if you can. Thank you.
I don't mind at all, not a bit. So on slide 15, you say you assimilate MLD and nutrient among other variables. Where do you get the data for MLD and nutrient time series from? So let's go back to slide 15. Okay.
Okay, so the mixed layer depth, I got it from the I guess, okay, so I got it from the NOBOM, I guess, NASA Ocean Biogeochemical Model, which gives an estimation of the mixed layer depths. Obviously, one can use any other estimate. This is not a prerequisite to use from any model. And what is the other one, you said? Mixed layer depth and there's something else.
Okay, the mixed layer depth and nutrient time series from. Where did you get the data?
Yeah, so both of them I considered from the NOBOM output. But in principle, if one want to use this method, they can use the estimates from any other observations or models as well.
Okay, thank you, Dr. Roy. There is a next question for you from Dr. Lerder, who spoke yesterday. He said, this is a very interesting approach to derive information on phytoplankton functional types. Do you think the satellite approach will work as well in coastal optically complex waters?
And that's a very interesting question, and thanks for finding this interesting. Ideally, the algorithms were developed for open ocean, where we have got less disturbances. However, if we can retrieve the coastal ocean chlorophyll and light absorption characteristics or light absorption coefficients in a better way, in a more refined way, then these algorithms can also be used there, because the algorithm assumes that the specific absorption coefficient and the chlorophyll concentration are more or less error-free. And if it has got error less than 30%, then the error in estimation is always less than 30%, as I showed in the last slide. So it's basically the data processing group's jobs, in a sense, to figure out the more refined values of the chlorophyll concentration and specific absorption coefficient from the coastal water.
Okay, thank you, Dr. Roy. And here's an additional comment. First I will read, and then I will move to the next question. From Jim Acker, for Adonis, some toxins like mercury do not go away, but actually propagate up the food chain so that large predatory fish have excessive concentrations. And then the next question is, does the model note any association between phytoplankton and higher sea surface temperature?
Well, that's a very good question. I actually didn't look at this aspect in a sense so far in this paper that I presented, but this is what I am doing at the moment. I don't have the results ready to present now, but I'm looking at how the SST variation is associated with pico, nano, and micro. So hopefully the results will come up after some time, but it's a very good question actually.
Okay, wonderful. Thank you. Are there any additional questions for Dr. Roy? We do still have time within the question and answer period, so I will wait a couple of minutes here to give Okay, the next comment is from also the workshop organizer, Dr. Acker, who said, you should see my talk this afternoon for empirical observations in Lake Erie.
Yeah, I'm waiting for that talk. I've seen the title and I'm really waiting for the talk.
He said, if it's not too late.
No, it's not too late. I'll be waiting for the talk.
Okay, and then the next question is, have you tried to compare your results to the method developed by Harada? I hope I'm saying that correctly at all.
Well, the PFT algorithms used by various other methods, not only by Hirata, there are many other PFT methods. We have been doing an intercomparison of the PFTs from all different methodologies, and that work is being done by a large group where I have given input from my method. So at some point we'll be having either a paper or something concrete to say how the different methods compare with each other. So because several methods are not directly comparable, so we are trying to figure out all these methods that gives, say, for example, phytoplankton size class, and then comparing those. And that includes the Hirata's method. Then there are a couple of others that include biogeochemical model. That some of them use the total chlorophyll. So there is an inter-comparison project going on, and I have given input from my model there.
Okay, so there's a follow-on. Is it part of the Marine Ecosystem Model Intercomparison Project?
Not really. This is basically done by a group from Hokkaido University in Japan, and also from the input from the bio-optical model developers. That includes the Plymouth Marine Laboratory and few other institutes.
Okay, thank you, Dr. Roy. Are there any additional questions? Let me scroll down to ensure that I haven't missed anything. Cecile says thanks for your response. Any additional questions from anybody for Dr. Roy? I'll give it just a minute or so to give individuals an opportunity to type. And if not, what we will do at that point is we will break for lunch. Lunch break today is again 2 hours long, so it will be from, in this case, closer to 11:45 a.m. Eastern Standard Time through 2 p.m. Eastern Standard Time, at which point we will resume the meeting with 2 additional talks for the third day of the workshop. Okay, and just a reminder, tomorrow the poster session will be online. Okay, and you'll see the URL for that later today as well. You will not be joining me in this meeting room tomorrow, okay? Okay, Jim's typing here. Let's see what he has to say. Any additional questions? Let's see. Jim said thanks again. Okay, so Jim Acker said email addresses for the poster session authors will be available on the webpage. So actually, let me go ahead and I will pull that up for you in just a moment here. And he's typing some more, so let me see what he has to say before we do that. Okay. He says this is a divergence from the previously announced access method that was emailing him. He would prefer that you email the individual authors regarding any questions for their posters, which will be posted at the URL. And let me just pull that up really quickly again. I think I've got it on the break slide. And you'll see that again later this afternoon. So it would probably be a very good idea to, if you go to the Goddard DAC webpage and the Giovanni portion of the page, you see that this is a listing within there, but it'd be good to bookmark this for tomorrow. And let me move next to the Q&A pod, since I have Dr. Roy's presentation available for you to download. Okay, and again, to download the file, you select the file and an opportunity to download appears after that. That's an option for you. Okay? All right, let's go ahead and we'll take our lunch break at this time. I will leave this up just for another minute or so, and then I'll put the lunch break information up. Again, we will not be monitoring the room during this time period, but certainly feel free to post any questions that you may have. The question and answer feature is persistent throughout the day, so that content is not lost. It will be maintained. We can certainly follow up with any offline discussions that need to take place by way of email if you think of something. All right, well, thank you, Dr. Roy. And did you want to then, shall I just move you down into the participant slot?
Yeah, and thank you very much, Jennifer, and thanks, Dr. Aker, again, for inviting me. Thank you very much.
You're very welcome. It was my pleasure. All right, thank you, sir. And everybody, we'll keep the telecon line open, but again, I will not be monitoring the room nonstop during the next 2 hours, although I